Hello everyone, I'm Xiaomin Lin from the Institute of Psychology, Chinese Academic of Science. Today, I'll present our recent work, a corticospinal signature for interindividual pain sensitivity. In this work, I'll show how we developed and validated a machine learning model that links brain and spinal cord data functional connectivity to predict pain sensitivity. Pain sensitivity is how strongly a person feels pain, like their pain threshold and pain tolerance. In everyday life, we can easily see even healthy people feel pain differently, even to a same pain stimuli, because pain is a complex sensation and emotional express. It's not just about sensing something, but also about how the brain and the spinal cord process this information. To study these pathways, we often use non-invasive imaging tools like resting state fMRI. While the brain is resting, it still runs preset networks called resting state functional networks, groups of regions whose spot signals rest and fall together. Dr. Biswal first found them in 1995, and many teams later confirmed famous ones like default mode network and or sensory motor networks. For years, we thought the spinal cord is too simple for this. You know, spinal cord is a small structure and a simple structure. But in 2014, my mentor, Dr. Yazhou Kong, first and his colleagues showed that the human spinal cord also has resting state networks. These findings push us to view pain processing as a full corticospinal system. Building on this, new imaging now lets us see brain and spinal cord together. Before, we scan them separately. Now, simultaneous corticospinal fMRI records both at once. Even at rest, we spot spontaneous resting state networks tightly linked between cord and cortex. Like in this study, the researchers found that some spinal segments even correlation with the brain's resting state networks, like the ventral halves of the spinal cord with the somatosensory networks of the brain. However, in this study, the authors only mapped the resting state networks of the brain and the spinal cord at a purely physiological level, and didn't examine how these networks related to behavior. By contrast, a paper published in the Nature Communication shows that the brain's resting state networks alone can predict pain sensitivity with good prediction and strong statistical support. Well, that leads me to ask, can combined corticospinal resting state networks predict pain sensitivity just as well or even better? And if so, is there potential for clinical application? And how much does the spinal cord contribute to those predictions? To answer these questions, we collected 6 data sets. 4 healthy volunteers trained and validated our model. We scanned them with corticospinal T1 and multi-band EPI sequence, measured the pain threshold. Finally, we tested our model on 2 clinical cohorts. The first one is diabetic patients with chronic pain, and the second one is doctor-associated pain patients. Next, here is how we answered 4 main questions. First, we built a machine learning model to predict each person's pain threshold from resting state corticospinal functional connectivity. We turned it with leave one subject out cross-validation in dataset 1 and tested it in 2 external healthy groups. Second, we carved out a pain sensitivity fingerprint, the corticospinal signature for pain sensitivity, or CSPS, using bootstrapping. Third, we compared 5 alternative models to show the integrated corticospinal connectivity is critical. Finally, we applied the CSPS to predict clinical pain in 2 patient cohorts. Well, here is our results. In dataset 1, leave one subject out cross-validation yield a good correlation of Pearson R equals 0.71 with actual pen sensitivity. We then tested the CSPS in two external healthy cohorts. The results showed statistical significance between CSPS response and the actual pen sensitivity in two healthy groups. Finally, dataset 4 involved 40 participants who underwent simultaneous cortical fMRI while receiving either a neutral cold stimuli or a heat pen stimuli. In this modality-specific test, CSPS correctly distinguished the pen in 74% of all cases. Well, CSPS comprised 29 non-zero predictive connectivity weights, of which 16 were positive, meaning stronger functional connectivity predicts higher pen sensitivity, and 30 were negative. The first positive connection is very interesting, which is the direct connection between the sensory-motor network of to the ventral horn of the spinal cord. In other words, we found that the direct functional connectivity between the primary motor cortex and to the ventral horn of spinal cord segment C4, which is related to pen sensitivity. Next, we asked whether pen prediction really needs both both brain and spinal cord signals. We trained five extra models on dataset 1. Model 1 here kept only brain connectivity. Model 2 only cord connectivity. Model 3 combines M1 and M2 features. Model 4 kept brain and cord separate by deleting every cross connectivity. Model 5 did the opposite, kept only the cross connectivity. When we tested them on dataset 2 and 3, neither brain-only nor spinal-only models generalized well. And that true predictive performance comes from the integrated corticospinal functional connectivity. Finally, we tested CSPS in two clinical pen cohorts. First, in diabetic with chronic pen patients and the subacute doctor-associated pen patients, CSPS responses correlated with their clinical pen ratings. When we combined both clinical cohorts, CSPS explained 32% of the variance, and the yield R equals 0.56. Importantly, CSPS outperformed all 5 submodels, and even the established models like the RPN showed in the figure, which is published in the Nature Communication. CSPS outperformed all these models across healthy and patient data sets, underlying its strong clinical applicability. Then we tested, did you remember our strongest functional connectivity is the primary motor cortex to the spinal cord? We tested the causal role of our signature's strongest link, the direct functional connectivity between M1 and the spinal cord. You know, because M1 is the most common target for pain treatment with TMS, we used RTMS to perturb that pathway in healthy adults. Specifically, we recreated some healthy participants. Each did a middle heat pen test and a corticospinal resting state scan before and after stimulation. By comparing CSPS response and pain ratings before versus after RTMS, we can see whether disrupting M1 causally changes both our signature response and actual pain sensitivity. Well, interestingly, these two changes were significantly correlated, meaning that participants with larger drops in corticospinal functional connectivity also showed larger shifts in their pain reports. This finding supports CSPS as a casual marker of pen sensitivity, directly tracking how M1 spinal modulation alters perceived pen. Before I finish, I'd like to acknowledge everyone in the lab for integrative neuroimaging of pen for their hard work and support. Special thanks to my mentor, Dr. Yazhou Kong, and the founding bodies that made this research possible. If you'd like to discuss the work further or see more details, you can email me, reach me on WeChat, or meet me at my poster 2044. Thank you.
Thank you, Xiaomin. Do we have any questions on the app?
There's one question.
Yeah, would you like to ask?
So this is quite a general question, I think. Probably doesn't apply just to your research, but to pain research in general. So it says pain is a highly subjective experience. How to assure that a self-reported rating is comparable across subjects and modality?
I think it's a big question because, to be honest, we have no way to make sure that the self-report pain rating is accuracy, but there is only one thing we can believe is self-report pain rating.
Maybe if I can rephrase it closer to your area, would your corticospinal signature for pain be reflective of nociception proper, or also incorporate some cognitive and emotional factors there? So do you think it is really doing something about the pain proper, the sensory input, or covers a wider range of?
We did some confident analysis, and we found our CSPS response isn't correlated with some, for example, emotional questionnaires or other maybe depression questionnaires. So we made a conclusion that our CSPS is pen specific, not others.
Thank you. Do we have any other questions? Maybe I can also ask you how you actually measure sensitivity, since we all were wondering what it incorporates.
Okay, sensitivity.
Or pain threshold. So what was the actual method of measuring that?
We use thermal pen stimuli. We put the thermal pen stimuli here.
Yep.
Then when the participant feel pain, they will click the mouse to stop the pen. We write down the temperature when they feel pain, and we will make this pen as their pen threshold, then pen sensitivity.
Yep. Thank you very much. Well, in the interest of time, we'll move to our next speaker, but let's thank Xiaomin first. Yeah.